exports smaller molecules, so they're antibiotics, toxins, you get toxin, these would be the kind of type 1 secretion systems that you see out of the cell. And again, it takes energy, because you gotta cleave the phosphate. There's a hemolysin protein that's affiliated with it, and some other, this high B protein that's again the trimer, the heterotrimer that makes a channel. So again, you have a lot of different components of the channel that actually either integrates in the membrane, that's a major part, or gets latches on and then moves it through. So that's kind of the whole thing. So here in type 1, I'm breaking down this diagram. This diagram in your book has all the secretion systems in one big panel. But just looking at type 1, so here's your cytoplasm, you make it, you know, blah, blah, blah. This is going to be like the SRP or one of the other TAP protein systems, and then it comes in and gets folded, and then you have this one, at least this one's called the Toll-C protein. Again, they're very specific, and it'll secrete, you know, the whatever the protein it is. So it makes these barrels, okay, polar on the inside of those barrels, but again, a channel that opens and closes, and then you just export it out. Okay, so the whole point of it is make the proteins, try to grab onto whatever protein you're specific to and make it go through. And this is a direct thing. It's a channel that goes directly from here to here, okay? There's no monkeying around in here, it just goes right through. And again, smaller molecules use this system right here. Okay, for 2, this is related to the pili assembly. So actually making pili, this is also, this type of secretion is actually related to how you secrete. Because you know, you think about pili, right? So flagella, all the proteins that are on the outside of the cell, they have to be exported, right? So let's say your LPS, a gram-negative bacterium, those LPS don't just pop out there, right? They have to be exported to be made on the outside of the cell. The same thing with the flagellar apparatus. All the proteins that make the flagellum have to be exported and then assembled, right? They have to be assembled, so you have to be able to do that. This one it's more complex. It's got 15 proteins, and it's embedded in the inner membrane. It's an inner membrane, so it kind of goes through. There's only one protein that is in the outer membrane. It forms a beta barrel, so again, it's a direct shoot between the inner and the outer. And then through the periplasm, there's a protein that's a gated channel, so it controls how much protein gets through there, through the 2 different gates out to the export, okay? And cholera toxin is one of these that uses type 2 secretion pathways. So when you look at it, this one in the beginning can either use YAG or TAP to get the substrates through the inner membrane, okay? So when I talked to you about these 2 different secretion pathways for the inner membrane, it could be down here, okay, and it depends on what it is. And this is just using one particular set of gated channels. But you can have either SEC-YAG, recognizing, you know, grabbing onto the signal sequence, coming over here, going through here, getting it folded, cleaving off the signal sequence, and then pushing it through here, all right? And then you get whatever the protein is made out here. So this is going to be at least using these 2 pathways through the inner membrane, getting it out. And again, you have this beta barrel set of proteins that secrete out to the outside. So that's just type 2. Again, it's all about making channels, right? And then type 3, so type 3 secretion system. This one is a syringe. Okay, it's kind of cool actually. It has 20 proteins, and this one really much is, type 3 is really affiliated with virulence pathways. So when you see nasty E. coli like EHEC or Shigella, a lot of them use type 3 secretion pathways to do this. So usually it makes the virulence defectors in the cytoplasm, you're making the toxins in the cytoplasm, and you have to export to the outside. And what it does, it's like a syringe because these pili land on the host cell, and they inject those toxins directly into the host cell. That's why it's like a syringe. So instead of just exporting to the environment, it targets a particular host, all right? And these are also known as the injectosome because it's injecting whatever it's exporting in there. Kind of cool. Enterotoxigenic, I think, I can't remember if it's toxigenic, EHEC or ETEC. One of those, what they do is when you land, I should show you guys the movie. It's a very cool cartoon movie, but when they land on the host in your gut, they attach to the inner membrane of your lower intestine. And the pili attach, and then they inject proteins that get your cells to make all this myosin filament. And so what it does is your cells start making all this really great filament so that the bacterium ends up pushing up against the host cell, and the host cell makes all these different filaments. It looks like it's standing on these big pillars, so it just grows. And that's your cells doing that, but the bacteria is inducing it because it's injecting these effectors that are making the eukaryotic cell grow. So whenever you see like an EHEC infection, all the E. coli are sitting on these little pillars in your gut. Really lovely thing, right? But it gives them more surface area to attach. So sneaky little buggers, they're just like doing that. So anyways, so what the injectosome does is it looks like this. So you have, and also, they're also related to the flagellum motor, all right? So remember that you have this whole cascade. Remember when you get some kind of solute that the cell goes, oh, okay. We instigate this whole cascade. Remember the methylation of the CHE? You guys remember this from 311? Kind of, maybe? Not really? All right. So remember you have the tumble or the run, right? So the tumble is when there's no solute around, you're just like tumbling around, and the flagellum is either going clockwise or counterclockwise, right? And so then when you get some kind of attractant like glucose, it says, okay, don't tumble anymore, go straight to the solute. And so what that does is it changes the direction of the rotor such that now they're going to go on a run instead of a tumble, okay? And the syringes actually are related to how these proteins are exported to make the flagellum, which is kind of cool. So not only is it used for virulence, it's also used for something quite good, which is motility. So type 3 looks like this. So you have your inner membrane transport proteins. So it's the proteins being made here, gets the signal sequence comes through, gets folded, gets exported here. In this case, it's a little syringe, all right? So it doesn't just go out. There's a little extra set of proteins that it injects that, whatever that protein is, a toxin, into the host cell where it's sitting on. So these will just land, these will land on the host cell and just inject those toxins right in there. So it's kind of cool. Like I said, EHEC does that really, really well, actually. So, all right. The other kind, type 4 now, this is, it's involved in DNA transfer. So this is conjugation, right? So you have your conjugation pili. These are also found in gram-positive bacteria and archaea. And when you look at the way these are set up, it's actually related to the plasmids. Well, you guys remember like eons ago in the lecture of self-transmissible plasmids. So remember, those are the plasmids that can actually help move themselves to a new host cell. And again, they take energy. So always these things, whenever you're moving proteins out of the cell, it's always going to take energy to do it. You got to get something moved across the membrane. You got to burn up some energy to do it. So in type 4, again, making your protein down here, all right? You got a bunch of different proteins. In this case, it's just using the genes. It also has a little injector-like thing, and it comes out and gets secreted out to the outside. So these can also be associated with virulence type pathways. These can be settling on a host cell as well. But again, when they look at the gene sequences, these are very similar to the pili made for conjugation. So this would be like a conjugation pili. And you're going to move DNA there. But in this case, it's moving protein instead of DNA. So you can think about when you evolve something like this, what comes first? Adapting yourself to move DNA to another cell in a conjugation pili, or then maybe co-adapting that so you can move nasty things out of the cell to a host, right? So it's using the same system, but you know, it's like, was it a pathogen first, or was it just trying to move its DNA around, and then it said, well, I can do these other things with the same apparatus. So bacteria are very good at kind of co-opting different mechanisms to do different jobs. So this is just this is the full diagram now in your book, and it's just giving you a variety of the different types, 1, 2, 3, 4 here, and just using a set of different operons to do different genes. Okay, these aren't just set in stone. It's just that you have to just remember direct. Okay, this one has kind of some other pathways here. This one is a syringe. This is more like a pili. That's a conjugation pili. So that's kind of what you just have to remember, is just the subtle differences between these two to get the protein outside of the cell. All right, type 5, they have their own beta barrels, okay? They're autotransporters, so they have 4 domains affiliated with that transporter. So they have a translocator at the carboxyl terminus that forms the beta barrel in the outer membrane. They have an adjustable linker that extends to the periplasm, and then they have this passenger protein that has some functional parts. I'll show you the diagram in a second. And then protease, again, cleaves off the passenger off the translocator. So again, this is like the signal sequence cycle of that protein. So again, it's just a matter of, here I am, move me through, you cleave off the signal, export out of the cell. This one has a 12-stranded beta barrel, and but you need chaperones in the periplasm to fold the protein and move it around for all this assembly, and it also costs energy. So again, all these require ATP. Usually they're related to some other secretion pathways like these, they call them two-partner secretion transferring systems that move really large toxins, or toxins that have lots of different subunits attached to it, because you can't just move that hunkin' big thing through the cell, right? You gotta assemble it and then move it. And then chaperones, again, also help kind of move these secreted pathways, especially when you're assembling things like pillins and whatnot to get them out of the cell. So here, this is kind of an example of type 5 now, okay? So this is what it looks like. You have your N-terminus, there's this protease domain, this is gonna get cleaved off, it activates, it cleaves itself, and then the carboxyl end over here. All right, so N-terminus. So here's your protein. This is gonna get secreted out through here, okay, so it gets, energy is gonna take it through here, and then it's gonna come through here. In this case, it goes through this little beta barrel, okay, so 12 subunits of beta barrel, comes around, and then this one will get folded outside after it gets exported out here. It doesn't get folded in here, it actually goes through this because it's too big. Like if you have a huge protein, if you fold it here, it's gonna be like, you can't get it through, right? So you gotta somehow figure out how to get it through in a long skinny strand of amino acids, and then fold it on the outside. All right, and then finally the last one, type 6, these are actually commonly found in marine and soil bacteria. They're actually the ones related to biofilm and cell-to-cell communication, interestingly enough. They're really big, so there's 21 proteins affiliated with that channel. And this is kind of really cool because when you look at the proteins, they look like a phage tail. You know, the tail is the part that lands on the cell and injects the DNA in there. So you're thinking, maybe the DNA was from a prophage that got stuck in the chromosome. And then they said, well, the bacterium said, we can still use these genes to make our own kind of secretion of pathway. Instead of going in, they're going out. So it's kind of crazy, and so there's, like I said, and that one was newly discovered. That's the most recent one that just recently got discovered, and they've actually been finding it in Colorado, which is very interesting. So, okay, so that's mostly gram-negative because you got to deal with two membranes, right? But now you have gram-positives, right? So you don't have to deal with the lipid bilayer, right? The LPS, and you don't have an outer membrane. You just have this really thick periplasm, right? So these guys, because of that, you don't need as complex of a secretion pathway. So they don't have either type 1, type 3, or type 4 secretion pathways, much more simpler. And things like diphtheria, Clostridium, they have these AB toxins. Remember, that's a heterodimer that are they self-translocate, which is kind of cool. And what happens is that the beta subunit of these toxins binds to the surface